Hello, everyone. My name is Caroline. I am president of Acm-W, which is the women's branch of ACM here at UCLA. And this is Uma. She is in charge of our Distinguished Speaker Series, which is a series where we strive to bring accomplished and prominent individuals within computer science here to UCLA to speak to all of you about their work and to share their knowledge with our community. Today's speaker is Professor Tandy Warnow from the University of Illinois. And then Uma is going to tell us a little more about her.
Yeah, so first of all, thank you very much for coming, Tandy. We are very honored and excited to have such a distinguished professor and speaker come and speak for our community. And just a little bit of a background, so Tandy, she did her PhD in mathematics at Berkeley, and then she did her postdoc at USC. And she has received so many prestigious awards, and she's a fellow of so many distinguished organizations, one of which is the ACM Fellowship. And that's just one of so many. So we really are honored to have you come speak for us. And yeah, we're excited to hear about this talk that you have. It's just a little bit of a general overview. It's kind of about the intersection of computer science, mathematics, and biology. So I think it's a topic that we'll all find very interesting. Yeah. Okay.
Thank you. So this talk is meant to be understandable to people who have no biology background. And I really want you to believe that. You don't have to know biology to follow this presentation. So if something comes up, just interrupt with a question. I don't need you to delay your questions to the end. So this is a picture of a bunch of birds. And the reason there are a bunch of birds is that one of the things I'm going to tell you about is research that we did in developing algorithms while we were trying to understand how birds evolved. So evolutionary trees are what I'm going to be talking about, and evolutionary trees of birds is one of the topics. So, by the way, my talk is on my webpage, so all of my talks are on my webpage, and you can actually download the talk, and you don't have to take notes, you can just go get it. Okay, so this is an evolutionary tree of human, chimp, gorilla, and orangutan, and what you can see is showing is that human and chimp have a more recent common ancestor. So how do we get these evolutionary trees? The way that people get evolutionary trees is they gather data. Typically, the data right now are genome data, so they're DNA sequences that they get from genomes, and then they apply software, okay? And they can treat the software as a black box, and then they get a tree. But what I'm going to basically tell you about is what's inside that black box. And what's inside that black box is really computer science and mathematics. So the background is biology, but what we're going to do is computer science and mathematics. So this is a cartoon about sequence evolution. So you can think of DNA sequences as just strings of A, C's, T's, and G's. And these strings are evolving from an ancestor. So imagine that this is the root of the evolutionary tree up there. And you're looking at the different genomes in the different species. And you're looking at a stretch of the DNA and just saying, how does this DNA evolve? And if you look at this tree, on different edges, things are changing. So on this edge, between here and here, the first letter is changing, right? There might be other changes as well, but the strings are changing. They're not changing length, but the letters are changing. So that's an evolutionary process. And in the end, you get the sequences start at the root with the ancestor, and they go down to the leaves. And in the end, you just get to look at the sequences at the leaves. So you're trying to reconstruct the tree from the genomes that you see. And you can think of this as just strings of A's, C's, T's, and G's, and you go from that to a tree. So the first thing to notice is that the tree that I'm drawing here has no root. Okay, again, a graph theoretic tree, it's a connected acyclic graph, right? There's no root here. This is not a rooted tree. It's an unrooted tree. And yet, what is generating the data is a rooted tree, right? There's a rooted evolutionary tree. We're using DNA sequences to get at that tree, and then we're constructing an unrooted tree. You always have to remember that we get unrooted trees, and that's basically mathematically, it's because the mathematical models are time reversible. So when we get a tree, it's unrooted. Just remember that. We're getting unrooted trees back. Okay. Statistical consistency is a statement about a statistical estimator, and you want the error in the thing you're estimating to go to 0 as the amount of data increases. So you can think of this as like if I have a coin and I want to know what is the probability of a head. If I keep tossing the coin, I can get a better and better estimate of the probability of a head. So my error will go to 0 as I get more and more data. And for me, data is coin tosses. In the context of phylogeny, it's sequence length. As your sequences get longer and longer, your error in your estimate of the tree will go to 0 if you have a good method. Okay? If you only have a single nucleotide, you can't tell very much. If you have, thousands of nucleotides, you should be able to get a correct tree with very high probability. So statistical consistency is a statement about an algorithm. The algorithm wants to have a guarantee that as the data increases, the error will go to 0. Okay? This is a concept from statistics. So what I'm going to describe for you is computer science and statistics coming together. Everyone okay with statistical? How many people have heard this phrase, statistical consistency? Just a few. Those of you who haven't heard it, just remember it's a statement about a method. The method error should go to 0. Okay. Since the amount of data impacts your accuracy and more data should lead to lower error, you would hope that genome scale data would let you get species trees correctly. Now, why do we even want species trees? It turns out that there's so many things in biology where knowing how the collection of species evolved helps you answer questions. And there's this famous saying from Dobzhansky that said, nothing in biology makes sense except in the light of evolution. So understanding the evolutionary relationships lets you answer biology questions. Things like protein structure prediction. Lots of things depend upon having a good tree, a good evolutionary tree. But I'm just going to talk about how to get that evolutionary tree. I'm not going to talk about the applications. Okay, so we want to use genome scale data to get at the evolutionary tree with this idea that more data are good, right? Everyone thinks big data is great, right? Enough data, you can answer everything. Okay. Genome scale data to get phylogenies, that's called phylogenomics. So phylo is for phylogeny, which means evolutionary tree. Genome scale data, genomics, phylogenomics. The thing is that we want to use the genome scale data to get at the evolutionary tree of different organisms, in particular human, chimp, gorilla, and orangutan. The correct tree should be human and chimp are siblings, right? Separated from gorilla and orangutan, which means there's somewhere to root that tree, and you would root it, in fact, on the edge going to orangutan. And then you'd get the correct tree. Okay, we'd like to get the genome scale data telling us the correct tree. We're going to look at this genome scale data and look at how recombination breaks up the genome. You don't have to understand what that means. You just have to know that there are different regions in the genome that are broken up by things called recombination. So these different regions within the genome are going to be called genes. They don't mean they have any functional meaning. They're just called genes. It means they're regions in the genome. Okay? The problem with genome scale data is, anyone know what the problem with genome scale data is? The problem with genome scale data is that they're not all the same. The different parts of the genome don't have the same evolutionary history. It's really bizarre, but different parts of the genome have different evolutionary histories. So it's not that you're getting more data and you can just use it. You're getting more data, but it's a mess. So you get this thing called genome discordance, gene tree discordance. So remember, the correct tree is supposed to be human and chimp, more closely related than they are to the other things. But if you look at the tree over here on the left, human is sister to gorilla. That's not what we're expecting to see, right? Human sister to chimp, that's what we expect to see. So the problem is different parts of the genome have different trees. And when different parts of the genome have different trees, you just don't know what to do. But this is real. This is not just a mistake. And the main cause for this heterogeneity is something called incomplete lineage sorting. So incomplete lineage sorting is a population-level process that makes discordance between different parts of the genome. And it comes about from a population-level thing where imagine that every row is a generation and every dot is an individual. And what you're doing is you're tracing your history from yourself to your parent and from that parent to its parent, and keep going backwards, tracing the history of that one allele. And eventually, everything has a common ancestor, so eventually all these lineages come together. But in this particular tree, although human and chimp are siblings, right, in the dark tree inside the broad stuff, the other two are siblings also, and they shouldn't be. So the true gene tree can be different from the true species tree. So what are the things that make for this to happen? So this mathematical model going backwards is called coalescent, the coalescent model. And what's happening is that at each point, you get to pick a random parent from the previous generation. So if the generation is large, you have a lot of options, okay? And in a particular edge, you only have a certain number of generations. So if you have a very small number of generations, and two of you are trying to find the same parent, so you coalesce in that edge. If you have a large population size and not a lot of generations, you might miss each other. And if you miss each other, you go back to the next edge above, and then you might randomly coalesce. And that's when you get a different tree. So that's what's happening here. These two lineages should coalesce on this branch, but they don't. And because they don't coalesce, all three lineages come up here, and then the other two coalesce first, and you get a different tree. So mathematically, this is not biology, this is mathematical. On a mathematical model about coalescence, gene trees will be different from the species tree with a probability that depends upon how rapidly you have speciation events. Rapid speciation leads to more incongruity, okay? So if you have genome scale data, some of the gene trees are going to be different from the species tree. And the more rapid the speciation, the more you're going to see that. So when you do human, chimp, gorilla, orangutan in particular, it's a problem, because it's really rapid radiation, and human and chimp are almost identical. So incomplete lineage sorting is what people are seeing when they look at their genome scale data, and it's confounding analysis for many biological data sets. And there's a very big debate in the literature about how to construct species trees when you've got this kind of heterogeneity. And roughly the debate goes like this. On the one hand, the traditionalists say, just keep analyzing your data the way we always have, use the same methods, and just get the tree that you get there because it's tried and true, okay? And then the other side says, but from a mathematical perspective, your model doesn't assume heterogeneity, it's not going to work, and we have evidence it won't work, so you should use our methods. Then the other side says, but your methods give bad results, and then the first side says, but you're just wrong because you just don't like our results. Okay, so it goes back and forth like this. So there's a big argument, but it really comes down to theory, what you can prove mathematically, and what you see on data. So it's an argument between theory and data, okay? And what I'm going to do is tell you about that argument, tell you about the evidence, and explain where it actually makes sense and where it doesn't make sense, and what we can do better. So you haven't asked any questions yet, and I'm hoping someone will ask a question at some point soon. Okay, so I told you that we did birds. So the Avian phylogenomic project, we worked for about 3 or 4 years together to do this phylogeny of birds, 48 birds, whole genomes. And we had this really interesting thing happen. We used the traditional method, which is just called concatenation. You take your genome scale data, you put it into one really big, long matrix, and then you run standard methods on it. And we got a tree that made sense, except that the tree had these really, really, really rapid radiation, right? And we know with rapid radiation that we can have a problem. And when we looked at the different gene trees, they were all different from the species tree. So we had a situation where we had a species tree we thought was reasonable, but we knew that it was a condition where we would have incomplete lineage sorting, and we had a huge amount of heterogeneity among the gene trees. And when we have that much heterogeneity, the traditional approach isn't supposed to work. So we went and took the best method that was available that took heterogeneity into account. And we got another tree that was a tree that did not make sense. So we had this problem where we had 2 trees. One was based upon the traditional approach, it made sense. The other one was based upon the novel approach, it didn't make sense. And the people on this project were arguing with each other about what to show. Should we publish both trees? Should we publish one tree? If we publish one tree, which one do we publish? It was a really big mess. Okay? So that's what I want to point out. I also want to say that our analysis took 250 CPU years. Only 48 species. These are computationally intensive methods. Okay, so another project, this time plants, more species, not quite the same number of genes, but again, massive heterogeneity that was consistent with a rapid radiation, i.e. incomplete lineage sorting. And at the time we tried to analyze this, the same basic time we were doing the Avian project, we didn't actually have workable software that could handle heterogeneity. The best method, i.e. the method that we used here, which was called MP-EST, and I'll show you data later, the method that was available for a smaller data set wouldn't run on this data set. There are too many species. So we had a situation with the plant project where we couldn't even use the available software. Okay? So to deal with this, we had to develop a new method. So it's the developing the methods that I'm going to tell you about next. So this talk, I'm going to give you the sort of the mathematical framework for the incomplete lineage sorting, the mathematical model for incomplete lineage sorting, and then I will explain to you how you can get statistically consistent species tree estimation even when you have massive heterogeneity. And then I'll tell you how we improved that estimation with new algorithms. So it's the improvement with new algorithms that's the computer science. Okay? And until I get to the point, it'll just be basic mathematics, probability theory type stuff. And again, you don't have to know any biology. Okay, so we're going to try to solve this problem. You know, human, chimp, gorilla, orangutan. We're going to look at genome scale data. And that means we're going to be sampling different parts of the genome, which I'm just calling genes. We're sampling different parts of the genome. We're going to try to get a tree from different parts of the genome. So remember I said that there's this mathematical model of coalescent? And this mathematical model gives you a probability for every gene tree. Okay? Every gene tree, this is one gene tree. Every gene tree has non-zero probability. So in fact, the species tree, which is a tree with branch lengths, right, for time and population sizes, every species tree gives you a probability distribution on gene trees. So we're going to use that probability distribution to get the species tree. Instead of treating it as a problem, we're going to use it as something that guides our inference. Okay? So statistical consistency is error should go to zero. We want methods that will have the theoretical guarantee of having error that goes to zero as the amount of data increases. Okay? The main competing methods, one is concatenate everything into one really big alignment and then just run your favorite method. And your favorite method here is probably maximum likelihood because it's a nice rigorous method. It's computationally intensive, but it gives good results. Even heuristics give good results. And the other approach are called summary methods. So summary methods look at the genome scale data and gene by gene, they get trees. So you get trees at different parts of the genome, and then you take those trees and you combine them. Okay? Two different approaches. One just concatenates everything into one big matrix and then runs your standard approach like maximum likelihood. The other one gets trees on all of the individual regions and then combines the trees. So the first approach of concatenation is the traditional way. And the second approach of getting trees on the different parts and then combining them, that's the new way. Okay? Quote unquote new way. So what do we know about the traditional approach? So we now know there's a theorem that was proven by Sebastian Ruch. By the way, he was here. Sebastian Ruch was here in the math department. So this is due to UCLA. Sebastian Ruch and Mike Steele from New Zealand proved that maximum likelihood, concatenation analysis using maximum likelihood, is not only not consistent, but it can actually converge to the wrong tree and give you 100% confidence in that wrong tree. So that's a really big problem about concatenation, that you can run your analysis and be really, really confident that you have the right tree because all the statistical support values are 100%, but you'd be wrong. Okay? So that's a problem with concatenation from a theoretical perspective. But from an empirical perspective, it's much more interesting. On simulated data where you're not violating the model, everything's under the correct model, you can get concatenation being better than statistically consistent methods. So it's not a black and white story. And understanding why under simulations concatenation is sometimes better than the right methods is part of the interesting thing here. Okay, so mixed accuracy in simulations. Sometimes it's worse, sometimes it's better. So this is an example where it's not consistent. You know, as you increase data, error is going down, but then it just sort of plateaus. So the data here are like the number of genes, right? You have more and more genes that you're taking from the genome, and if that data is going to infinity, you'd like your error to go to 0, but it's not going to 0. It's gone down, but then it's just hovering. So what methods are statistically consistent? Nothing that you would think about. Not concatenation using maximum likelihood. Not the most frequent gene tree. So what do I mean by that? You've sampled all of your genes, right? And you've gotten trees on all of your genes. And then you look at them and you say, well, which one happens the most often? I'll just go with that. Not a good choice. That can give you the wrong tree. With probability going to 1. I mean, that's a big problem. It can give you the wrong tree. All sorts of consensus methods won't give you the right thing. So nothing that anyone was coming up with was working. Okay? And in particular, I want to talk about this most frequent gene tree thing not working. So there's this thing called the anomaly zone. The anomaly zone is exactly the case where the most probable gene tree is not the species tree. And what is now known is that for whenever n is at least 4, there are rooted species trees where the most probable rooted gene tree is not the species tree. And when n is at least 5, there are model species trees where the most probable unrooted gene tree is not the species tree. So 2 things. All this is saying is that there are anomaly zones. There are cases where the most probable gene tree is not the species tree. But they start at certain values, depending upon whether it's rooted or unrooted. So in particular, for the rooted case, if you only have 3 species, this one doesn't say whether there's an anomaly zone. The next theorem says there are no rooted 3-leaf anomaly zones, which means if you only have 3 species, the most probable rooted gene tree is correct. And there's no anomalous 4-leaf unrooted gene trees, which means if you only have 4 species, the most probable unrooted gene tree is correct. Okay, most of you, if not all of you, are computer scientists. Most of you have taken an algorithms course. How do you use this to get a species tree on 100 leaves? So, the challenge is here. I'm going to give you, let's say, 1 million different gene trees on 100 leaves. And they all have the same set of species, same set of 100 leaves. How would you figure out the correct species tree? You can do, if we're going to do rooted gene trees, right, then every 3 you can solve. So you can get the correct species tree on every 3 leaves. If you had the correct species tree on every 3 leaves, can you get the correct species tree on everything? Find a sibling pair, right? Throw out 1, recurse. Insert. For 4 species, it's the same story. So summary methods that are going to combine gene trees. For rooted ones, for every 3 species, look at all the gene trees, take the most frequent gene tree you see, and if you have enough gene trees, the most frequent one will be the most probable one, and so you won't make a mistake. And if you don't make a mistake on any 3, then you can combine them by finding sibling pairs and recursing. And the same thing is true for 4. What I've just shown you are polynomial time, statistically consistent methods for getting species trees. And the reason that this is important is you just have to believe it's doable. Okay? So theoretically it's doable, but there's something really wrong with both of these methods. They're statistically consistent, they're polynomial time, but they will have bad performance. Why? Anyone? Because they're going to fail almost all the time. Why are they going to fail? If they make a single mistake, if a single tree, a single 3-leaf rooted tree or a single unrooted 4-leaf tree is wrong, they won't succeed in putting the tree together. So they're very fragile. It's a proof of concept, but it doesn't work. Okay? They need extremely large amounts of data to be correct with high probability. So, statistically consistent under this incomplete lineage sorting condition, you need to have methods that are not so fragile. Statistical consistency is easy. Good performance on data is hard. Okay? This is the thing about computer science. This is not just statistics, it's actually computer science. You need methods that are robust to models that you don't have perfect data. So there are lots of methods that are out there that actually take advantage of these ideas of rooted 3-leaf trees, combining them, and unrooted 4-leaf trees and combining them. Okay? And they've become very popular. MP-EST does rooted ones, Bucky does unrooted ones, and there's other methods. And all of these are statistically consistent. Some of them are polynomial time. So I haven't mentioned co-estimation. Co-estimation is input is just your sequence data, and then you co-estimate the alignments. You co-estimate the gene trees and the species trees. And that's even better. Co-estimation has even better accuracy than combining gene trees, which I'll show you in a few minutes. But that's another category, and the reason it's not so common is it's too expensive. So these co-estimation methods just take too long. By the way, those also come out of UCLA. So the co-estimation method that is the most well-known actually doesn't come out of UCLA. There's another co-estimation method that comes out of UCLA. I can't give the credit to everything to UCLA today, sorry. But anyway, Bayesian co-estimation of gene trees and species trees is very powerful, but does not work well in large data sets. Okay, data. This is a simulation study where we took species trees with only 11 species, and ILS, so there's gene tree heterogeneity, not a huge amount, but there's gene tree heterogeneity. And on the x-axis, we increase the number of genes. So we start off with 5 genes, and we go up to 50 genes. And we're looking at different methods, and we're measuring error on the y-axis. So the error is the proportion of the branches that you get wrong. So you want that proportion to be 0, right? You want to get 100% accuracy. So what you see is that on 5 genes, there's high error for everything, right? High error for everything, then lower error, then everything's perfect. Okay, so in here, the red and yellow have the best accuracy because they have the lowest error. Red is star beast. It's doing co-estimation. Yellow is concatenation using maximum likelihood. Does anyone remember what I told you about concatenation? It's not statistically consistent. It has the best accuracy at 5 genes. It ties for best at 10 genes. It's perfect at 25 genes. Concatenation is better than some of the other methods. In fact, concatenation is just better than the other methods. The other methods include methods that are statistically consistent. This is an example where concatenation is better than the statistically consistent methods. And this is one of the reasons that not everyone has jumped on the bandwagon of doing it a new way. They want to do it the old way. Concatenation is not bad. But star beast is quite good, but it's not even as good as concatenation. So here are some observations. Star beast is more accurate than the summary methods, and what I'm not showing is that the reason that it's more accurate is that when we run star beast, we get better gene trees. So star beast co-estimates gene trees and species trees. It's getting better gene trees. And then if we take the gene trees that star beast gives and we run them through one of these summary methods, we get a perfectly nice species tree. Star beast is better because it gets better gene trees. Summary methods are statistically consistent, but this simulation is not giving them true gene trees. It's giving them estimated gene trees, right? This whole simulation is you get a species tree, you get gene trees, you simulate sequences on the gene trees, you estimate gene trees, and you combine the estimated gene trees. What do you know about statistical consistency? The guarantee of statistical consistency assumed true gene trees. There's no theorem about estimated gene trees. So this is a complex protocol, and you're combining estimated gene trees. There's nothing golden here. So concatenation is giving the best results, but it doesn't even have any nice theory. And if we look at one of these statistically consistent methods, in this case MP-EST, and we give it estimated gene trees, we get about 8% error. And then if we give it true gene trees, we get 0 error. Estimation error in the gene trees is why it's doing badly. So the first thing for you to notice is that all of this argument about statistical consistency is assuming true gene trees, and you never get true gene trees. Has anyone here done any phylogeny estimation ever? No. It's the kind of thing you never get perfectly true trees. You always have error. Even if it's only 5% or 10%, there's error. Okay? You don't get correct gene trees. There's always some error. So this is the main thing. This is the main problem, is that the approach that is trying to get species trees by combining gene trees has to deal with estimation error in your gene trees. The gene trees have estimation error, the species trees will have estimation error. And this is where the debate has really focused. You have people who are arguing in favor of one approach that assumes that you have true gene trees. You never have true gene trees. Okay, key issue in the debate is gene tree estimation error. So back to the Avian project. So we had, 48 birds, whole genomes, and we had this big problem where our species tree using concatenation looked good. The species tree using MP-EST looked bad, right? And we had this realization that it might be because of gene tree estimation error. And that's why we were getting results that didn't make sense. So that led us to do a different analysis. So we had this concatenation tree that made sense. We wanted a coalescent-based analysis that would also make sense. We needed to get better gene trees. So how do you get better gene trees? You have genome scale data. How do you get better gene trees? You have this concept of statistical consistency, which is as you add data, error goes down, right? You've got all these gene trees. Maybe some of the gene trees, actually, the estimated gene trees are different, but maybe the true gene trees aren't different. So maybe you can look at your sequences, your genes, and you can cluster them. And here we're back in computer science world, right? Cluster the genes so that within each cluster they have the same evolutionary tree, the same history. If you can do that in a rigorous way, then you can combine the data for each cluster and get a better gene tree. What I didn't perhaps say is that on this data set, the average branch support in our gene trees was 25%. So those of you who know something about phylogenies, and maybe none of you do, 25% is really bad. You really want to have about 90% support. 25% means that probably only 25% of your edges are correct, which means you have really, really, really uninformative data. You don't have enough signal. So you have two different gene trees, but it's just because you don't have enough signal to get a real gene tree accurately from them. So we wanted to get better gene trees, and we came up with this thing called statistical binning. So statistical binning was also published in Science, and it's basically a graph algorithms problem. Okay, so you might see this picture down there where it says incompatibility graph, and that graph has vertices with colors. So it's a vertex coloring problem. So what we did is we said we have 14,000 different genes. We're going to make a graph out of these 14,000 different genes. Each gene is a vertex. We're going to put an edge between two genes, i.e. between the vertices, if we think they have different trees. And the way that we do that is we look at the statistical support on their branches. So now you've got this graph where you have edges between vertices, meaning that they should not be in the same cluster. And then you want to cluster these vertices. That means you're assigning colors. Vertex coloring, so you never put the same color at the end of an edge. So that's a proper vertex coloring. And you want to find, let's say, a minimum vertex coloring. Everyone here knows that minimum vertex coloring is NP hard. So we used a heuristic, and we separated the vertices into sets so that every set, the vertices have no edges between them. And that means we can put them together into one set, one long alignment, and we can get a new tree. Okay? We have more data for each gene. So that's the idea behind statistical binning. We want to get better gene trees. So we're taking advantage of this idea of concatenation is not bad. We can bin things together. So that analysis, statistical binning, gives you new gene trees, which we call super gene trees. And those super gene trees are then combined instead of the original gene trees. Okay? But you'll notice that there's only one gene for every color. Whereas we started off with one super gene for every color, but we started off with a lot more genes. So we made a version of this called weighted statistical binning, where we have as many super gene trees as we had original gene trees. Okay, so we're just replacing the original gene trees. Just a minor modification, but that minor modification gives a nice theorem. Gives a theorem of statistical consistency. Okay. Here are some data showing the impact of statistical binning. So what I'm comparing, the y-axis is error, and the x-axis are just different methods. The red version of each method is what you have on the original gene trees. The blue version is what you have on the new gene trees. In every case, the error is going down dramatically. So by partitioning the genes into sets in this automated way, you're getting better quality species trees. Error goes down across the board. It also improves everything else we looked at. Branch length estimation, the incidence of highly supported false positive branches. The gene tree topologies I've already shown get better. I didn't show that. Gene tree topologies get better. The species tree topologies get better. Everything gets better. Okay? It's almost magic, right? We're getting better gene trees, so we get better species trees, we get better branch lengths. So here's a comparison on some avian data, a simulated data set, where we're looking at what happens concatenation compared to binning and unbinned versions of MP-EST. And that's on this left-hand side. So this in black is concatenation. This is the unbinned MP-EST. And these are the binned versions. So what's happening on the x-axis is you're making the sequences longer. You start off with short sequences, then you make them longer. As you make the sequences longer, the gene trees that you start with are better. Okay? If they're very short sequences, then they're very bad gene trees. As you make them better, then you get better gene trees, and the binning doesn't help. What you can see is that unbinned is red. It's much worse than binned, right? So binning is improving the species trees. Concatenation starts off here, so it's better than unbinned, and then it's worse. So this combination of statistical binning is better than both concatenation and the unbinned version. It's not just better, getting close to concatenation. It's actually better than both. Okay? So statistical binning is improving species trees, and it's actually better than concatenation and better than the original way of analyzing your data. And the only reason is that it's giving you better gene trees. Okay, remember I started off by saying that the avian analysis, we had two trees, and we couldn't make sense out of them? These are the two trees. So, more or less the two trees. So, everything that's in red is something that conflicted with the concatenation analysis. Everything else is no conflict. So this is what you get when you use binning with MP-EST. This is what you get when you use no binning. If we had put together this one, the unbinned version, we would have gotten a tree that had strong conflict with the concatenation analysis. But by binning, we eliminated the conflict. So it's just showing you that you can actually have a big impact on a biological data analysis this way. Any questions about this? So in the end, this is what we published. So in the science paper, we published this the statistical binning version of MP-EST, which is here, along with the concatenation analysis. So then we didn't have conflict. We started off with conflict, we came up with a better method, and then we didn't have conflict. Okay, running time comparison, 250 CPU years for the concatenation analysis, 5 years with the new analysis. So it's a big reduction in running time. Okay, so the summary so far. Statistical binning improves gene trees. Improving gene trees leads to improved species trees. And that's basically the story. And it improves the running time. So that's statistical binning. I just want to remind you that statistical binning, the point was to get better gene trees. We needed better gene trees so we can get better species trees. Of course, you want gene trees for other reasons. But statistical binning was a combination of a graph algorithm, which was an NP-hard optimization problem for which we designed a heuristic, and using statistical ideas to do the decision about how to do the partitioning, okay? How to design the graph. Okay, the second project I'm going to tell you about is plants. And when we got to plants, we could not run MP-EST. MP-EST was too slow. So we had to come up with a new method, and we came up with something called Astral. So Astral is based upon the observation that there are no anomalous four-leaf unrooted gene trees, which actually means that you can do quartet-based species tree estimation. So you take all of your gene trees, and now you're going to compute the score of a species tree by looking at 4 species at a time and saying, does it agree with the gene tree? And just sum up all of the counts. So 4 species at a time. This is a discrete optimization problem. Okay? You don't have to know any biology to think it's just a discrete optimization problem. It's a median problem in trees. You're constructing a tree to maximize the similarity or minimize the distance. It turns out that this, if you can solve this exactly, you have a statistically consistent method for species tree estimation. However, it's NP-hard. So we came up with a constraint version. And the constrained version becomes polynomial. So the original problem is NP-hard. The constrained version is polynomial. The constrained version is, I still want to optimize the score, but I'm going to give a certain set of allowed bipartitions. So every edge in a tree gives you a bipartition on the leaf set. And I'm going to tell you which bipartitions you're allowed to use. So if I tell you which bipartitions you're allowed to use, I still want to find an optimal solution. That constrained version of the problem is polynomial. And it can be solved using dynamic programming in quadratic time. And it's still statistically consistent if you draw the bipartitions from the gene trees, and then you can add to it. So this is a simple, discrete optimization problem, statistically consistent, polynomial time. How well does it work? We did a massive simulation study. We compared it against MP-EST, and this is tree error on the y-axis. And this is what happens as you increase the number of genes. So when you start with only 10 species with 1,000 genes, with 10 species, they have the same accuracy or the same error. At 50 species, there's a big difference, right? Because blue is what Astral is. At 100, there's a huge difference. And at that point, you can't even run MP-EST, but Astral keeps running, and it can go to 1,000. So 1,000 species with 1,000 genes, and it still has good accuracy. And it's fast. So it can complete, this is 24-hour analysis, and it can run on 1,000 species and 1,000 genes. And this was an early version of Astral. It's actually a lot faster now. It can also handle other causes for gene tree discord, including horizontal gene transfer. It's still the most accurate of all the methods. This is, Astral is in red, and the y-axis is as you increase the amount of horizontal gene transfer. So you have 2 causes for gene tree discord, and Astral is still better. And actually, the interesting thing here is due to UCLA. Sebastian Rock proved that under a random model of horizontal gene transfer, this method, Astral, will be statistically consistent. So you have this really nice, simple algorithm that's only based upon quartet trees. There's nothing about biology there. There's nothing about statistics there. It's just saying the quartet median tree is the method you're trying to solve within a constrained search space. And it's statistically consistent under various different models, and it's highly accurate. So what that tells you is that you don't have to do messy parametric Bayesian analyses to have high accuracy. Okay, biological data. This is an interesting plant data set where Astral and MP-EST, which are two different coalescent methods, give different results. When the MP-EST analysis was done originally, they came up with an analysis that was different from the traditional concatenation analysis for this particular data set, in particular where Ambarella went. And they said, it's clear that there's incomplete lineage sorting. MP-EST takes incomplete lineage sorting into account, therefore we're right. But Astral also takes incomplete lineage sorting into account, and it actually matched the concatenation analysis. So what that's telling you is that when you want to design a method, it's not enough to know the conditions under which it's statistically consistent. You actually have to look and see what happens on finite data, because relative performance depends upon the conditions of the data, not just upon what is motivating the method. So Astral's used all over the place at this point, and it's largely because it's fast and highly accurate. So the summary, Astral is a statistically consistent method. It combines gene trees. When the gene trees have estimation error, there are no guarantees about statistical consistency, but it has very good performance in practice. And it's actually now the thing that's actually replacing, complementing concatenation analysis. So it's become the dominant species tree method when there's incomplete lineage sorting because of its accuracy so far. But the main thing is this gene tree estimation error impacts accuracy, and we don't really understand theoretically why. I mean, we know that it does, but we don't have any kind of guarantee. So there's a basic theoretical question, which is what is the impact of gene tree error on species tree estimation? So basically we know that if you have true gene trees, that as the number of gene trees increases, your error will go to 0 if you have good methods. But what if you have just a small amount of error, like 1% error in your gene trees, and then the number of genes goes to infinity? Can you prove anything? And right now we have no proofs of anything positive. The only positive things we have are basically that if you have a strict molecular clock, you're fine. But if you don't have a strict molecular clock, you're not fine. And no one really believes in the strict molecular clock. So we have a situation where we really don't have any positive theory when there is estimation error, which presents a basic statistical question. How do you design methods when you have to take into account estimation error in your input? Right? Your input is not perfect. You'd like to model that error, but it's really hard to model that error. So fundamentally what you have is you have no positive theory. All you have are simulations and theory that applies to idealized conditions that you never face. So the argument between the two domains, you remember I told you they keep arguing, right? The argument, I think, is largely in favor of the people who are dubious about theory. And speaking as a mathematician, that's disappointing. But what it means is that we need new theory and we need new methods. So future directions. We need better coalescent methods that are more robust to gene tree estimation error. We need methods for co-estimating gene trees and species trees that are actually scalable, because right now they're not. We need better theory about the impact of gene tree estimation error. Like maybe we could say, if your error follows this model, that with enough data, you'll have predictable amounts of error, and you'd be able to locate where that error is. That would be nice, but we don't have any such theory. So better mathematical theory would be good. And then there's this entire new class of methods which don't fall into the category of either concatenation or summary methods. What they do is they look at the concatenated alignment, and they just look at each of the sites. And each of the sites they're looking at taking heterogeneity into account, and they're figuring out the entire species tree without ever going through the gene tree calculation. So that approach they're called single site methods. They're very promising. They bypass gene tree error, and they take heterogeneity into account, but so far they're not as accurate. So what I would summarize this by saying is that This is a very hot area in computational biology. It's an area where mathematics and computer science have a lot of things to contribute, lots of low-hanging fruit. Even just speeding up the Bayesian methods would be a big contribution. And there's high impact because the number of phylogenies that are built every year is enormous. The number of species trees that are built every year is enormous. And new methods that can give better accuracy would get used. This is not a saturated field at all. So, the tree of life has multiple grand challenges. I've only been talking about one, but everything in that list is worth working on. How to get large-scale trees, how to get trees on millions of species, how to get multiple sequence alignments, how to get trees without going through multiple sequence alignments. All of this is available using different kinds of computer science and math, graph theory, probability theory, hidden Markov models. It's all there. And none of it actually requires background in biology. So for those of you who are interested in doing algorithm design and having an impact on the scientific world, I encourage you to look in this area because there's a lot that can be done without becoming a biologist. So the things in particular that I really like working on, ultra-large multiple sequence alignment, that's what I talked about here a few weeks back. Supertree estimation, really clean combinatorial optimization problems. And then these mathematical guarantees under Markov models of evolution if you're mathematically inclined. So the other thing to say, big data are not just massive amounts of data, they're actually different. As you take more and more genome scale data, you actually have different kinds of data. You have slower rates of evolution, you have noisy stuff, you have model heterogeneity. See, that challenges everything you're doing. And here are my acknowledgments. Siavash on the left, he developed all the methods, and he's now at UCSD. So just down the road. Baiza did a bunch of the stuff. Bastien Bousseau did a bunch of the theory. And then Sebastian Roch, originally from here. Thank you. Okay, so there hasn't been a single question, and I'm hoping there's one now.
So, how, in the case of animals, you trace back their ancestors? Is it just the degree of coincidence of the genome? What criteria are you using?
Are you talking about how do we get the species tree, or how do we get the gene tree, or?
The gene tree, where you're trying to trace the family tree. Who their ancestors were and so on.
So let me just go back to the very beginning. Okay, so the classical phylogeny problem is you have DNA sequences that have evolved down a tree, and you're trying to get the tree. You take a mathematical model of evolution, and you look for the maximum likelihood tree. So it's not like you're looking at traits. It's not like you're necessarily trying to minimize distances and go backwards. It's really a statistical estimation problem. So maximum likelihood under a mathematical model of evolution. And that's how you get the gene trees. And then the species tree, you can do it by combining gene trees or just treat all of the genes in one big matrix. Jason.
Do people look at other units besides genes to estimate the species tree, like units that would take into account either genic and chronic regions or do it at an exon level?
So in the bird phylogeny, the things that I call genes, some of them are exon regions, some of them are intron regions, some are UCEs. But so it's really just, it's not necessarily strictly speaking regions, so much as parts of the genome that might be concatenated together. So, but there's still different regions in some sense within the genome.
And how do they decide on those regions?
Typically using traditional criteria that have to do with you're looking for a particular, what is normally called a gene, and then you're taking that gene region and dividing it into introns and exons and stuff. But the quote-unquote right way to do it isn't what people do in practice. Yeah.
my understanding, when we're building the gene tree itself, we would need the genes or the sequences of the genes of the population that was like before the split between the species. Is that correct?
No, you don't need the ancestral sequences.
But like we still somehow like trace them back. Like, can you like explain?
So let me. That's the point.
Basically, just like the simplest way to build a gene tree.
Well, that's the same question that was just asked. So the situation here, we have 5 sequences that are the input, and the output is a tree with 5 leaves, right? So the input is sequences that are going to be at the leaves only, and the method for coming up with that tree is you come up with this. So there are some standard models for sequence evolution, and like the standard one, for example, Jukes-Kanner, says that the sequences evolve down a tree, and then it's worthwhile looking here, under a model where every edge has a probability of substitution on it, and if you change, you're going to change to the other nucleotides. So what you're trying to find is a model tree with those substitution probabilities on the edges. And that maximum likelihood problem is how you construct the tree.
So we're basically just coming up with the genes that we did not have, but we're saying that the better the tree, the less substitutions we have to make.
That's parsimony. That's parsimony. So I just want to point out that what was being asked is, are you looking for the tree that has the smallest number of substitutions, right? That's a very famous problem called maximum parsimony, and it's not exactly the same as maximum likelihood, and it's another NP-hard optimization problem. So it is actually one of those methods that's not provably statistically consistent, but has good accuracy a lot of the time. So the construction of gene trees is itself like a big research area. But what I've been talking about here is assuming you've already got the gene trees, how do you get the species tree? And I skipped everything about how to get the gene tree, right? But gene tree estimation, there are books and books and books on this. And I just said, let's take that as already solved and look at the species tree problem. Yeah?
When you do your statistical binning technique, are you sort of effectively, once you color your graph and pick some genes to bin together, you then effectively build a new gene tree, treating that pile as one big gene?
Absolutely. That's exactly what you do. And one of the details that's important there is, now suppose you put 7 different genes into the same bin. So these genes, what you did is you assumed that they had you did a test to figure out that they might have the same tree topology. That doesn't mean that they have the same numeric parameters. So what you do when you're doing the tree estimation on that bin is you say, look for a single tree topology, but allow each of the genes to have their own numeric parameters. So that's called a partitioned analysis. So, that's one of the ways that we get around the fact that they are going to be different, but there's only a finite number of different tree topologies. So we're putting them together because they might have the same tree topology. Yeah?
So follow up. They can extract gene sequences from fossils.
Yes.
So there is knowledge of going back all that time. It's not just hypothetical or extrapolation from the present. There is knowledge about that, and that's a way to verify, of course, that this is good.
I don't think you can use it to verify, but what people do when they have fossil data, it's not usually genome scale because it degrades, but they will use the fossil data in their input, and those sequences are part of the input, and then they go at the leaves of the tree, but because they know that they're fossils, they actually sort of push it into the tree. So they just collapse the edge that it's connected to. So the fossil data can help you under some circumstances, but it's still not the dominant way that people analyze data because there's not enough high-quality fossil data. But fossil data is more useful for dating, which I don't discuss. Did you have another question?
I mean, sort of a related question. When there's these disputes in the field and methods coming up with a novel finding, how is there sort of buy-in from the community to believe this method versus another method? Is there some sort of orthogonal validation? concatenation done or something.
The problem with evolution is you can't know if you're right. But I think one of the things that we keep finding with statistical binning and with Astral is that the similarity between the trees that we get and the trees that concatenation gets tends to be very high, which tends to make people comfortable with both analyses, right? So when you see similarity, you're less worried about it. When you see differences, you don't know what to do, right? And the other thing is that the field does depend upon simulations. So this is because you can't know if you're right, doing extensive simulations under a wide range of conditions is how people test their methods. So Astral has been tested and tested and tested, and it really matches or improves upon the competing methods. So that's why Astral has been taken up. The Astral is now dominantly in use. Statistical binning less so, and I think that's partly because it's actually, statistical binning is this weird thing where you're both doing concatenation and you're doing a coalescent analysis. And the community tends to be divided into, they either want one type of analysis or they want the other. But when you do a statistical binning, within each bin you're doing concatenation, which makes the coalescent people crazy. And then you're combining them using a summary method, which makes the concatenation people crazy. So it's like, you know, it's not pure. Any other questions? Yeah?
One more last question. So, yeah, you perfectly answered the previous one. So I understood that you were mainly talking about like how we combine them, but like the metrics of how we decide like which gene trees do or not, in my head. But I think I have a question. Oh, okay, great. It might be something related to what the other person has asked before, but when we have a tree and then we try to like merge the trees, do we just treat the tree as a single genome that was doing, or no?
So when you say you're trying to merge the trees, are you talking about the binning technique? Or you're talking about the Astral thing?
Well, I think you mentioned that you combine the trees like in multiple methods.
Oh, the summary methods. So the combination, it's not that it's combining the trees, it's that you're using the collection of trees to get summary statistics, and you use those summary statistics to find another single tree from all those trees. I'm just emphasizing that you're using the trees as your input. So you're not using the sequence data as your input, using the trees as your input. So, I mean, the particular technique that Astral does is it looks at the quartet trees in each of these trees, right? And it just tries to optimize quartet similarity. So that's what I mean. It's not really...
Like a structure. It's not going to be based on anything that's in the root.
No. And the trees are unrooted, right? These trees are unrooted. So there's like nothing there except for their shapes. Are there any other questions? Okay, well, thank you very much.